Hello. Today we'll talk about starting with the pancreatic hormones. So let's take some definitions of the pancreas. So actually, the pancreas is a mixed gland. A part of it, or majorly, exocrine, and there is small part that is endocrine. The exocrine part, of course, is responsible for secreting of the digestive enzymes, while the endocrine part, which is our concern in this lecture, produce several important hormones. The famous one of them is insulin. So as you can see, the pancreas is situated just beneath the liver, and it have the duct which pours into the duodenum, the starting of the small intestine, actually the duodenum, for the digestive enzymes. Looking closely inside the exocrine tissue part, it consists of a grape-like appearance of clusters of secretory cells, which is called acini, in plural, acini, and they secrete their secretion into a duct that soon unites these ducts and pour into the duodenum. While for the endocrine tissue, it consists of isolated islands of endocrine tissue called islets of Langerhans. There are around 1 to 2 million islets of Langerhans and represent around 1 to 2% of the pancreatic tissue. So more closely into the pancreas, we will find that the endocrine tissue consists basically of 4 types of cells: alpha, beta, delta, and the PP or F cells. The major one, of course, is the beta cells, which secrete majorly the insulin and another hormone which is called amylin. While the alpha cells, as you can see from the figure, the alpha cells, which represent around a quarter of the cells of the endocrine tissue, it secrete glucagon, and the delta cells secrete somatostatin. You can see that's a diagrammatic sketch for the different types of cells. And these delta cells secrete somatostatin, while the F cells or PP cells secrete a minor hormone which is called pancreatic polypeptide. And you can see the difference generally between the endocrine cells and the exocrine acini or acinar cells, that you can see that the acinar cells is kind of like surrounding the endocrine cells. And for sure, the endocrine cells need to be reside near to a capillary, because you know the endocrine pour its hormone or secrete its hormone directly into the blood. So for the pancreas, actually it do two major functions, either increase the blood sugar level or decrease the blood sugar level. And we'll see here in this slide what is the factor that affecting the blood glucose level or concentration. Of course, the first factor which increase the blood glucose level is the glucose absorption from the digestive tract. And another factor which is subject to hormonal control to maintain the blood sugar level is the hepatic glucose production. So once the body needs glucose, through the process of glycogenolysis and the glucogenesis, it breaks down glycogen and form glucose. While the factors that decrease blood sugar level is, of course, the transport of the glucose from the blood into the cells for either for the use and the production of energy or for storage as glycogen or triglycerides. Also, the urinary excretion of glucose, which can occur abnormally when blood sugar level becomes so high and exceeds the reabsorptive capacity of the kidney during urine formation. This urinary excretion of glucose definitely will decrease the blood sugar level in the blood sugar level. So let's talk about insulin, one of the major hormones that is secreted by the beta cells of pancreas. So it's one of the most important hormones that could utilize and coordinate the utilization of fuels by tissues. Its metabolic effects are mainly anabolic. So, for example, is the synthesis of glycogen, triglycerides, and proteins. Insulin basically composed of 2 peptide chains, peptide A and peptide B, and they link together by 2 sulfide bonds. If you can see, they link it together by 2 sulfide bonds, and as well there is additional disulfide bond is formed in the A chain. So you can see the A chain have its own sulfide bonds, and it consists of 21 amino acids, and the B chain, it's around 30 amino acids, and both A and B are connected by 2 sulfide bonds. So for the secretion, this slide show normally when the insulin is secreted, and it correlates with the amount of glucose that the blood glucose level. So you can see insulin release is not continuous. It's not continuous even after a meal, but oscillates with a period of around 3 to 6 minutes. So it's like bursts of insulin release. And this is oscillation. Oscillation means getting high, low in concentration of insulin. It's very important to consider when anybody is using is taking insulin as a medication, because actually the insulin had to be monitored to be taken in the right time during the oscillation period of the oscillation time of the insulin. So basically insulin is administered, and the oscillation is the target, not the constant high concentration. So you can see the blood glucose level. You can see this is the same timing with the insulin level concentration in blood. And you can see at the time of the breakfast, just as the time of breakfast, with the increase of glucose due to breakfast, the insulin burst and have a high concentration and then go back. And this is what we say the oscillation. So it oscillates with the amount of glucose that enters the body. And you can see as well that the insulin, it never shuts down. So it have like a minimal basal level of formation, the insulin. So what about the biosynthesis of insulin? Actually, the insulin gene in beta cells is transcribed into messenger RNA that is translated into single chain, which is called pre-proinsulin. And you can see the pre-proinsulin, it have, this is the chain, this is the A chain, this is the B chain, connected together by a C chain, and all the polypeptide have a signal peptide, and the signal peptide is very important in the trafficking of this precursor hormone. We'll see later. So the pre-proinsulin carries a signal peptide that directs it to the rough endoplasmic reticulum. This pre-proinsulin is modified by cleavage into proinsulin, in which the signal peptide, as you can see from the figure, the signal peptide have been cleaved. And this proinsulin is produced inside the rough endoplasmic reticulum. And actually it's obvious because the signal peptide, which already was present in the precursor hormone, have done its job in recruiting this pre-proinsulin into the rough endoplasmic reticulum. Once it is inside the rough endoplasmic reticulum, it is cleaved, this signal peptide, and two disulfide bonds will be formed between the A chain and the B chain. Once the proinsulin in this structure will be formed, it will be transported to the Golgi apparatus or Golgi network. In the Golgi network, two kinds of enzymes work on the proinsulin. It's the endopeptidase and the exoprotease. And these endopeptidase and exoprotease will lead to cutting this C chain to form a C peptide, which is a connecting peptide, and the mature insulin. And this insulin will be packaged into secretory granules and accumulate in the cytoplasm, waiting for the signal or the high blood sugar level in the blood, which lead to its secretion into the blood. Okay, this figure actually describes what we have just said. So initially, the DNA is got transcribed into messenger RNA, and this messenger RNA is recruited to the endoplasmic reticulum, in which the proinsulin is converted to proinsulin. Once it is converted to proinsulin, it is transported from the rough endoplasmic reticulum through vesicles and got transported. This proinsulin is transported to the Golgi apparatus or the Golgi structure. In the Golgi structure, we understood that the proinsulin now is cleaved by endopeptidase and exoproteases to release the C-peptide and the mature insulin that is packaged around the vesicles. And this proinsulin, as we said, cleaved to produce insulin plus C-peptide and packaged inside the vesicles and got released, okay? In the meanwhile, the glucose, which is outside, due to the effect of insulin, will be translocated inside the cell via a special kind of receptor, which is called GLUT2, glucose transport receptor, GLUT2, and it will transport it to the inside, where it undergo glycolysis by the mitochondria to produce energy, and as well it leads to the activation of two important pathways, which are the PI3 kinase and ERK1 kinase, ERK1 and 2 pathways. After the mitochondria is after do the job of glycolysis, of course, the ratio of ATP to ATP will increase. This will lead to the translocation of the potassium to the outside and the calcium for the inside. And calcium will aid more in the process of the exocytosis of the insulin. While the activation of the PI3 kinase and ERK1/2 pathway will lead to activation of transcription factor involved as well in insulin transcription and ERK1/2. So that's mean they add up the signal and amplify the signal in case that the amount of glucose in blood is so high. And this signal, it keep on going in a positive feedback until the amount of glucose in blood is lowered, and there will be a negative feedback to shut down the process of starting from the beginning, the process of transcription of the insulin gene. Again, we can see a more illustrative figure about what is happening. So initially, as we said, the gene coding for the insulin is transcribed to messenger RNA in the nucleus. And this messenger RNA is translocated to the cytoplasm, and by the help of the signal peptide, it will be recruited to the rough endoplasmic reticulum, where a translation initiates on the ribosomes. And they will form an N-terminal hydrophobic signal, which is a signal peptide. As we said, it aids in the transport of the messenger RNA to the rough endoplasmic reticulum. Once in the rough endoplasmic reticulum, you can see that the translated precursor hormone is inside the lumen of the rough endoplasmic reticulum. This N-terminal signal peptide, as you can see, it is penetrates. You can see it penetrates inside the rough endoplasmic reticulum, and the elongation of the polypeptide will result to the formation of the hormone. And soon after that, the signal peptide is cleaved, as you can see, and the proinsulin now is formed. And this one is the proinsulin now is formed inside the cisternae or the lumen of the rough endoplasmic reticulum. Soon after that, this proinsulin is packaged inside the vesicles, that soon these vesicles merge and transported to from the rough endoplasmic reticulum to the Golgi apparatus or Golgi complex, where it is further cleavage to form the connecting peptide or C-peptide and the hormone or the mature hormone. Soon after that, the insulin and the C-peptide are packaged into secretory vesicles that are released by exocytosis, releasing both the C-peptide and the insulin to the blood. So how the insulin secretion is controlled? To stimulate the insulin secretion, actually the main cause of stimulation is the elevated blood concentration of glucose, is a major signal for increased insulin secretion. This elevated glucose not only stimulates insulin secretion, but also transcription of the insulin gene, as we have just seen in the previous slides. So the glucose, the blood sugar level as well have a positive impact on the transcription of the insulin gene. Some neural stimuli increase as well blood glucose concentration of other few molecules, including amino acids and fatty acids, and these as well promote insulin secretion. The gastrointestinal hormone, like the intestinal peptide secretin, as well as the gastrointestinal hormone, stimulates the insulin secretion. These hormones are released after the ingestion of food. So they cause a rise in the insulin levels in the portal vein before even there is an actual rise in the blood glucose. So actually this is like a first line of to detect the increase in blood sugar level before being sensed by the brain. So what about the inhibition of insulin secretion? So the synthesis and release of insulin are decreased when there are a shortage of dietary fuels and also during periods of trauma. These effects are mediated primarily by an important hormone, which is epinephrine, the adrenaline, which is secreted by the adrenal medulla in response to stress, trauma, and extreme exercise. The stomach hormone ghrelin as well inhibit insulin secretion. So what about the mechanism of action? The effect, receptors, and the mechanism of action. So as we said, the main targets of the insulin, actually the liver, the muscle, and the adipose tissue. These are the main target. With the exception of brain, liver, and the erythrocytes, tissue require membrane glucose carriers. So that's mean for brain and liver and erythrocytes, glucose can enter and exit very easily. They don't need a glucose carrier, and it is obvious for these three major types of tissue or cells. But for others, if for instance it wants to enter the muscles, it needs a glucose carrier for it. So the insulin receptor actually it consists of two alpha subunits and two beta subunits linked together by disulfide bonds. As you can see from the figure, these insulin receptors, it's actually a monomer, and you can see the insulin is not bound yet. These alpha chains are only extracellular, and these alpha chains have the binding site for the insulin, while the beta chain, you can see that it can penetrate through the plasma membrane. The insulin receptor is have built-in kinase activity because it's a tyrosine kinase. And you can see it functions in transforming the phosphate signal, of course, as a kinase from ATP into a tyrosine residue. The binding of the insulin to the alpha subunit leads to the auto-activation of this insulin receptor, and actually it is auto-phosphorylated, thus activating this part of the receptor, which is the catalytic activity, or activating the catalytic activity of the receptor. And this activated receptor then phosphorylate another number of intracellular protein, which in turn affected the cellular response to insulin. So it's kind of like initiating the signal of the responses to insulin. So let's see the physiological effect on the carbohydrate metabolism. So the principal product of carbohydrate digestion and the principal circulating sugar is the glucose. And this glucose, actually the insulin facilitate the entry of glucose into the three major tissue, mainly the muscles, adipose, and as well liver. So terms used in carbohydrate metabolism include, of course, and you are familiar, I guess, by these terms, glycolysis and the glucogenesis and the glycogenolysis and the glycogenesis. And actually it is meaning the different terminology used in the process of metabolism of the glucose. So the binding of insulin to the receptor on cells lead rapidly to the insertion of the glucose transporter in the plasma membrane. That gives the clue that these glucose transporters actually already made, its proteins actually already made and stored inside the cells. And once these sense and they got the order that they need to internalize the glucose from the blood sugar, from the blood, they got recruited to the plasma membrane and open up for collecting all glucose that is outside, and this giving the cell an ability to efficiently take up the glucose. When blood levels of insulin decrease and the insulin receptor are no longer occupied, and the glucose, these glucose transporter which was recruited to the plasma membrane are recycled back into the cytoplasm. It should be noted that here that there are some tissues do not require insulin for efficient uptake of glucose. And of course, as we said, like the brain and the liver and red blood cells, of course. The transport of glucose from blood into different cells is done by, as we said, GLUT or glucose transporters, except a few tissues that glucose can enter cells without these transporters. So for GLUT actually there are seven isoforms of this protein have been well characterized, and each GLUT have been evolved a different mechanism. So actually it is responsible for a different tissue. So as you can see, the figure shows how GLUT4 is working. So insulin, it's a basic mechanism. Insulin binds to the monomeric receptors, leads to the alpha subunit, as we said, leading to the dimerization and activation of this tyrosine kinase, which is upon activation, it leads after a cascade of signaling protein and activation, leading to gene expression and regulation, growth regulation, and this signal transduction will lead to the translocation of GLUT4 into the plasma membrane for the internalization of the glucose molecule that soon will be utilized either for making glycogen or formation of lipid or for protein synthesis. So for GLUT1, 2, and 3, they are not affected by insulin. GLUT1 transports glucose across blood-brain barrier, while GLUT2 for kidney cells, while GLUT3 for neurons. And this is very obvious because they are not affected by insulin, because of course neurons and kidneys for the process of excretion and the propagation of nerve impulses, it don't need to be switched off. So if there is an insulin existing, it might switch off the process of free absorption from the kidney or the propagation of the nerve impulse. As well, brain needs glucose all the time, so insulin have no effect on these GLUT1 or 2 or 3. Therefore, in all these tissues, the glucose entry is insulin independent, while GLUT4 is insulin dependent and occurs in muscles and adipocytes. And these cells, as we said, these cells maintain a pool of GLUT4 in vesicles in their cell cytoplasm. In other words, these types of cells make GLUT4 already ready in the cytoplasm, and once it's as bound to vesicles, and once it is needed, it is recruited to the plasma membrane for the uptake of glucose. So you can see here, let's see what's happening for the process of recruitment. So you can see here, starting the insulin here bind to the receptor, to the insulin receptor. Initially, the glucose transporter are stored here inside the cytoplasm in the form of a membrane vesicles. Once there is insulin bound, and interact with the receptor, these vesicles move to the surface and diffuse with the plasma membrane. And this will increase the number of GLUT4 transporter in the plasma membrane. And you can see these small dots are actually the glucose molecules. And it starts to take up this glucose and open inside to release it to the inside. When the insulin level drops, that means the blood sugar level now returns to normal, these glucose transporters are removed from the plasma membrane by the famous process of endocytosis, forming again the small vesicles. The smaller vesicles fuse together to form a large endosome. And finally, these patches of endosome are enriched and full of glucose transporters, bud off to form small vesicles ready again to merge with the plasma membrane when the levels of the glucose, when the levels of insulin are high in the blood. Again, this figure shows the same process. You can see here, starting with the binding of insulin to the receptor, this binding lead to positive feedback towards the recruitment of the GLUT4 transporter into the plasma membrane, that upon fusion with the plasma membrane, now the GLUT4 is ready for uptaking and internalization of the glucose molecule. And when the insulin level decrease, the glucose transporter are recycled back inside the cytoplasm as a storage pool, and they can be recycled to do the job again upon the increase of the level of the sugar in the blood. So here we'll see, continuing the more elaboration on the carbohydrate and try to see the general picture. So actually insulin stimulates the liver to store glucose in the form of glycogen. So you can see here that insulin actually make the muscle promote the muscle for glucose uptake and inhibit glucose production from the liver in order not to add more glucose into the circulation. And as well, it inhibits the process of lipolysis, which lead to a formation of fatty acid. As well, as we have just said, that the insulin is secreted by the pancreas, and any beta cell dysfunction will lead the inappropriate secretion of the insulin. Of course, from the liver and the muscles, the blood glucose level can be increased. So the plasma membrane of liver is actually permeable to glucose, but still insulin increase the uptake of glucose by liver cells by increasing, of course, based on the concentration gradient of the glucose, and as well by enhancing the process of glucose phosphorylation and glycogen formation. As he said, and maybe you are familiar, because glucose, once internalized inside the liver cell, it is transformed into glucose 6-phosphate. And this step is very crucial and very important to keep the glucose concentration gradient outside is higher than inside. Because once it is converted to glucose 6-phosphate, the cell understands that it is a different molecule. So always glucose will be higher to the outside, and it can be go outside. And the glucose 6-phosphate will not return back to the outside. So actually insulin has several effects on in the liver, which stimulate glycogen synthesis. It activates a number of enzymes like glucokinase and inhibit the activity of glucose 6-phosphatase, and also activate the enzyme phosphofructokinase and the glycogen synthase. And all these enzymes actually work coordinated together to form glycogen. So the effect of insulin in liver cells actually increase the glycogenesis and glycolysis and decrease the glucogenesis and the glycogenolysis. And the insulin as well facilitate the entry of glucose into muscle, adipose, and peripheral tissue, because as we said, the membrane of adipocytes and the muscle cells are impermeable for glucose. So as we said, this slide summarizes the effect of insulin on the liver, and mainly the activation of a set of enzymes that lead by the end of the storage of the glucose as a glycogen inside the liver. So continuing about the carbohydrate metabolism, okay, so throughout the day, the muscles use the fatty acid. This is because under resting condition, the cells are dependent on GLUT4 for glucose uptake. But with a moderate or severe exercise, there are special GLUT4 vesicles, what we have just mentioned in liver, which is present only in muscles, move into the cell membrane in response to exercise only and do not require insulin. That's why exercise can lower blood sugar levels. So normally above the 20 age, at fasting condition, in fasting, the blood sugar level is around less than 100, while before meals it ranges from 70 to 130. After meal, maximum for 2 hours, it is less than 180, while before exercise, if taken insulin, at least it will reach 100. At bedtime, maximum by 140. So this is the levels of the sugar throughout the day. So let's talk now about the lipid metabolism. So insulin actually promotes as well synthesis of fatty acid in the liver. And when the glycogen accumulates to high levels, actually it's the maximum level of the maximum mass of liver that can occupy glycogen is actually 5% of the liver mass. So further synthesis is strongly suppressed. So once the liver reaches its threshold, the 5%, there is no more glycogen is synthesized. So, and the fatty acids start to take place, fatty acid synthesis starts to take place, which are exported from the liver as a lipoproteins. Any additional glucose taken up by the hepatocytes is shunted into two pathways leading to the synthesis of fatty acids. These lipoproteins enter circulation to adipocytes, which of course is used to form triglycerides. The insulin as well enhances this enzyme, important enzyme, which is the lipoprotein lipase, which activate the breakdown of triglyceride into lipoprotein to be used in the adipocytes. Insulin inhibits the breakdown of fat in adipose tissue by inhibiting another important enzyme, which is hormone sensitive lipase, HSL, that hydrolyzes the triglyceride to release fatty acids. So the insulin facilitates the entry of glucose into adipocytes, of course used to synthesize glycerol. And once a glycerol with the fatty acid from the liver is used to synthesize a triglyceride inside adipocytes. And you can see from the figure in the adipocyte, the glucose, once internalized, it's converted into glucose 6-phosphate that can lead to formation of fatty acid and then triglyceride by the help of the lipoprotein lipase and by the internalization of the triglycerides as well from the circulation. So again here, let's see what's happening. So we have the intestine. We have two major molecules coming from the intestine, the glucose and the fat. For the glucose, it goes as well the normal way. It's going through the under the effect of insulin. It is, of course, we are talking now in case that the liver have reached its threshold of the storage of glycogen. So it is converted to acyl coenzyme A, to fatty acid, to triglyceride, to very low density lipoprotein that is released back to the circulation. For the fats, it is transformed it formed the chylomicrons, and these chylomicrons is soon transported under the effect of insulin as well into the adipose tissue, where with the help of the very low density lipoprotein, it formed the fatty acid that soon is converted to triglyceride. So actually the liver makes as well VLDL cholesterol and releases it into the blood stream. So let's see here the mechanism. So insulin binds to the receptor, activate the PI3 kinase, which activate the AKT PKP protein kinase. This AKP protein signals through FOXO because actually inhibit the FOXO1. So what's FOXO1? These FOXO1 have a major role on the formation of the glucose 6-phosphate that lead to the glucose production and the hyperglycemia. Actually, that's the phosphatase, and activate as well MTP, which lead to the VLDL-GG TG triglyceride production, just we have said, and this will as well will lead to increase in the triglycemia in the blood, leading to hypertriglyceridemia. Okay, so both of these insulin will lead to hyperglycemia and hypertriglycemia. Of course, we are saying that when the threshold of the liver have been reached. So glucose doesn't depend on GLUT4 for entry into the liver. And the insulin has no effect on glucose uptake by the brain. We kind of like summarize major points. So the brain uses only glucose as its energy source. So it is important that blood glucose level is maintained above a critical level. So because a brain will sense any disturbance in the glucose, and it needs a certain level of minimum level in the blood to be working. Brain is permeable to glucose and can use it even without the effect of insulin. When blood glucose level fall too low, reach around 50 milligram per 100 ml, the symptoms of hypoglycemic shock develop. And this will lead progressive, of course, it will affect the brain and the nervous system, because as we said before, that the nerves have a type of GLUT transporter, GLUT3 actually, which is independent of the insulin. So this lower, very low blood sugar level will lead to hypoglycemic shock, and which will soon develop into progressive nervous irritability that lead to fainting and seizures and coma. So what about insulin degradation? So why there is insulin? Normally, why insulin is degraded? Because actually it have done the job, and there is no reason for staying in the blood. Otherwise, it will continue to do its action, and it will take up all the glucose inside the blood to either store it or promote its burning in muscles. So when glucose levels are very low, and this like a signal as in fasting state, the balance is pushed toward the intracellular protein degradation. So it's a kind of like a negative feedback for the glucose level in the blood. Actually, insulin is degraded, and its half-life time is 5 minutes in human, very fast, within the liver and the kidneys. So it is degraded by insulin in liver and kidney. This degradation of insulin involves endocytosis, internalization of the insulin receptor, the one which is responsible for binding to the insulin. So it's kind of like shutting down the door of the insulin to the cell by the action, of course, of insulin degrading enzymes. The major enzymes for insulin degradation is the glutathione insulin dehydrogenase, which cleaves the disulfide bonds. And of course, this disrupts the binding between chain A and chain B. And these are degraded soon after that. Each chain are degraded by insulinase. So let's see here a summary of the figure. So the blue arrow, this one shows increase, and the red arrow shows decrease. Once insulin binds to the receptor, this leads to as we said, the internalization of the glucose will increase. The internalization will increase. And of course, glucose 1-phosphate is made. And as you, if you remember this, and after that, the glucose 6-phosphate, of course, this will enable, this will maintain a concentration gradient that is high outside to the inside. This will use for energy utilization, okay, and the glycogen synthase starts as well increase, and this will lead to increase to the amount of the glycogen that is stored inside the cell, okay, and of course the amount of the glucose once this one is increased, the amount of glucose that is internalized will decrease. The process, once this happening, the process is, if the internalization of glucose is decreased, the process of glucogenesis will decrease as well. This lead to urea excretion decrease. As well, which add up to this process in this stem of the diagram shows when the amount of glucose is low, so the cell turn to take its majorly its energy from other molecules like protein. and as well amino acid internalization increase for making proteins. Lipid as well increase by the effect of if the lipase is decreased activity. So this will release to fatty acid into the circulation, and of course will form ketone bodies, decrease. Okay. So as a summary, so we have carbohydrate metabolism where glucose transport increase across cell membrane, glycolysis, and as well the activity of two enzymes, hexokinase and phosphofructokinase activity increase, glycogen synthesis increase, and the glycogen breakdown decrease. For lipid metabolism, the process of lipolysis decrease in adipose tissue, and hence the plasma fatty acid level decrease. The fatty acid and triglyceride in tissue increase. The uptake of triglyceride from the blood into adipose tissue and muscle increase, while the fatty acid oxidation in muscle and liver decrease. For the protein metabolism, actually the transport of some amino acid, as we have just seen from the figure, into the tissue increase. So the protein synthesis in muscles, adipose tissue, liver, and other tissues will increase. This will lead to decreased protein degradation in muscles. Okay. And we can see here you have a blood vessel, and you can see here that the pancreas have two types of cells, alpha and beta. It's a summary as well. Alpha cells secrete the glucagon, beta cells secrete insulin directly to the blood. And here you can see the glucose can, under the effect of insulin, it either do glycogen through the process of glycogenesis, or once the glucose level is low, the process is reversed through to be a glycogenolysis. And of course, these all are under the effect of insulin. So insulin promote the glycogenesis while inhibit the process of glycogenolysis. And this glucose as well can do the process of glucogenesis through the formation of fatty acids. And with a glycerol, it will form triglyceride. And you can see as well that insulin interact with this process as well, as well affected the internalization of glucose. It affected the process of the conversion of glucose into fatty acids, and inhibit the other process of conversion of glycerol into glucose. This in the liver. While in the adipose tissue, you can see that insulin promotes the internalization of fatty acid into the adipose, in which these fatty acids is transformed into triglyceride under the effect of insulin, and the process of lipolysis inhibited as well under the effect of insulin. While in muscle tissue, you can see the insulin promotes the internalization of amino acids and the glucose inside muscle tissue. The glucose is used up by the help of insulin and through the process of glycolysis to form ATP, and as well insulin promotes the protein synthesis and inhibits the process of proteolysis inside the muscles. So you can see here the action of the insulin on muscle cell. It promotes glucose uptake, glycolysis, and the glucogenesis, and amino acid uptake and protein synthesis, and it only downregulates the process of glycogenolysis. So it's actually a good slide summarize the effect of insulin on different tissue. So here another slide that shows what insulin can stop and what insulin can promote. And actually all of this we have just mentioned, except for this one, that insulin as well increases the uptake of ions, especially the potassium and the phosphate ions. And this is one of the important slides that show the diverse functions and regulations that is undertaken by insulin. So let's see now the factor that affect the insulin secretion. And actually you can see that all of them are opposing factors. So either release, stimulate insulin release, or inhibit the release. Of course, the factors that stimulate the insulin release is the glucose and amino acid and free fatty acids which are high in blood. The GIT hormone we have just said, like gastrin and secretin. The parasympathetic stimulation, acetylcholine, and some of the kinds of drug like sulfonylurea drugs. While the factors that inhibit insulin release is of course the decreased blood sugar level, the fasting, somatostatin, and the sympathetic stimulation by the epinephrine. And one of the important hormone is leptin, which control the fat deposit inside our body. What about the effect of insulin deficiency? So the first effect will be hyperglycemia, which is abnormally high blood sugar level, glycosuria, and that's excess sugar in the urine, polyuria, which actually the excessive passage of urination as around 2.5 liters per day for an adult, polydipsia, which is excessive thirst, polyphagia, which is excessive eating, and weight loss. And all these, together with the increased plasma cholesterol and phospholipid concentration, all these are marking of mark the insulin deficiency. So let's talk about diabetes mellitus, one of the major condition or cases that may arise as a disease due to insulin deficiency. So the first type of diabetes, actually, it's type 1. It's insulin-dependent diabetes mellitus. In this type 1 diabetes, actually, it is an autoimmune disease, and it is genetic actually, mainly, and some environmental factor may affect the occurrence of this case. It's characterized by severe, it's a very severe form of diabetes mellitus, and usually starts in juveniles and occasionally adult. And actually insulin absent because you have an autoimmune disease. So actually the beta cells that secrete insulin, it's got destroyed by the immune system of the body. And of course the plasma glucagon will be elevated. The etiology, actually the pancreatic B cells unresponsible to insulin, and they are damaged pancreatic beta cells due to autoimmune response and due to some of the environmental insults. Of course, their genetic etiology is involved in the progression of this disease. The symptoms, of course, as all symptoms of diabetes, is increased thirst and urination, constant hunger, weight loss, bedwetting in children, blurred vision, extreme fatigue, and irritability and mood changes. What's the replacement therapy for IDDM? According to promptness, duration, and intensity of action, so it's a case by case. So there are three types of therapy or drugs. There are fast-acting drug, which is actually zinc insulin, and there is an intermediate acting, like the isoflurane insulin suspension, and actually it is modified from the initial one, the fast-acting protamine zinc insulin, and you have the long-acting hedge-dome insulin zinc protamine complex. So actually these three different therapy for type 1 diabetes, and actually it had to be prescribed based on the case of the patient. Okay, so the doses as well is different based on the patient. So that's why you have three types of therapy based on the action of the insulin, if you need it fast acting or intermediate acting, or you have long acting insulin. So you can see there are some devices as well, new mode of treatment. This like infusion, you just have it in your hand or got attached to your belly, and this one will sense the blood sugar level and will like inject the correct amount of insulin, and you don't need to worry about when to take insulin injections. So actually we will see for type 1 diabetes, which will lead to a metabolic change, which is hyperglycemia and the ketoacidosis. So in hyperglycemia, it's caused by the increased hepatic production of glucose, as well. This is combined by the decreased utilization of peripheral glucose. So normally in healthy individual, so the pancreas produce insulin. This is the green structures. And this insulin moves glucose into the cells, okay? So, or promote the transportation of the glucose inside the cells. That's in healthy individual. While in the process of diabetic, the pancreas produce less insulin, so you will have more glucose in the blood, so less insulin will be moved inside the cells. So here in diabetic ketoacidosis, this glucose will be transformed. You will have ketone bodies or acetone bodies, which can be detected easily in the blood. So this ketoacidosis results from the increase of the mobilization of fatty acid from the adipose tissue, combined with accelerated hepatic synthesis of ketone bodies or acetone bodies. And because the lipoprotein degradation catalyzed by the lipoprotein lipase in adipose tissue, which is low in diabetic patient, the plasma chylomicrons of fat and VLDL levels are elevated, resulting, of course, to hypertriglyceridemia. So what about type 2 diabetes? Actually, type 2 is non-insulin dependent, and it's characterized by, it's a milder, of course, of type 1, and usually in adult, occasionally in juveniles, and it is heterogeneous group. So etiology, it's a subnormal level of insulin, and there are tissue insensitivity to insulin, and the pancreatic beta cells is non-responsive to insulin as well, and there is hyperglycemia, which impairs or affects the beta cell response. The symptoms looks like the same as type 1 with the addition of dry mouth and headache. And actually, it is due to type 2 diabetes. It's due to the combination of the defective insulin secretion and as well the defective response to insulin. So that means it might be the problem in the insulin receptor, which binds to the insulin. So cells will be either resistant to the insulin or have reduced sensitivity to insulin. And in early stages, the insulin sensitivity is reduced, which leads to hyperinsulinemia. And this severe complication of type 2 diabetes may result from the unnoticed type 2 diabetes, may lead to renal failure or vascular diseases like coronary heart diseases, and may lead to vision damage. That's why a lot of patients that have uncontrolled type 2 diabetes can lose their vision if they are not controlling their sugar level, or even they don't know that they have diabetes. So let's talk about the treatment of type 2 diabetes. Actually, diet is the first thing. You need to watch out your diet. Of course, insulin treatment or oral hypoglycemic agents is used when diet control is insufficient, and is used as well with insulin to lower the dosage of insulin. Drugs like the first generation of sulfonylureas, it is actually works to stimulate insulin release from beta cells, and they have adverse effect associated with cardiovascular disease and hypoglycemia. The second generation of drugs used in type diabetes 2 is another generation of sulfonylureas, and actually it stimulates the insulin release from beta cells, but as well release a glucagon and the somatostatin, and inhibit the process of hepatic glucogenesis, and enhance the sensitivity of the insulin receptors. And it is less, its side effects is less severe than the first generation of the drug. You can see these are some links for videos about the insulin and the mechanism of action and the effect, okay? And this concludes our lecture today about the insulin.